In this protocol, we introduce an in vitro reconstitution platform that mimics the lipid environment of mitochondria in the membrane. This platform can be used to investigate molecular mechanisms of mitochondria in the membrane fusion. The main advantage of this technique is that it allows quantitative investigation of integral membrane proteins and membrane-associated proteins in a near-native environment.
To begin, mix solutions A and B according to manuscript directions. Then generate the lipid mixture by adding the calculated volume of storage solution into amber vials with a glass syringe. Match the final volume by adding extra chloroform into the vials. Bake microscope cover glass slides at 520 degrees Celsius for 30 minutes. After baking, cool them to room temperature. Add approximately 10 grams of sodium hydroxide to 500 milliliters of methanol while stirring. Stir for 2 hours, continuing to add sodium hydroxide to the solution until precipitates start to show. Clean the glass slides in 10% sodium dodecyl sulfate solution, methanol saturated with sodium hydroxide, and 50 millimolar hydrochloric acid, sequentially bath sonicating the slides under each condition for 30 minutes. Clean the glass slides in ultrapure water for 10 minutes between each condition. Store the cleaned cover glass sealed in hydrochloric acid solution for up to 2 weeks to ensure good bilayer quality. Clean the polytetrafluoroethylene trough of the Langmuir-Blodgett dipping system using chloroform and ultrapure water until no wetting is observed. Spray chloroform.